And so today, I'm going to share with you our focal story of the invasion seagrass of Halophila stipulaceae. So where it came from, how it invades. But before we talk about invasion, I want to take a moment to appreciate what we're talking about. This is a mixed seagrass meadow with Thalassia testudinum and other mixed species like Serengonium filiforme. And it's one of our native seagrasses here in Puerto Rico, just swaying so peacefully. And these beautiful meadows are underwater powerhouses. Their roots and rhizomes stabilize sediments. They help protect coastlines. I'm not sure why this is timed, but I'm sorry. They protect coastlines. Their canopies create habitat and nursery grounds for fish like barracudas and snappers, as well as invertebrates like conchs and lobsters. And they're foundation species, and they create habitat and important connections between coral reefs and mangroves. And my favorite part about seagrasses is that they protect against pathogens. So here I'm showing you a study done by Lam et al. By 2017, where on the x-axis you have shore, flat, and reef. And on the y-axis, you have mean enterococci, which is fecal matter. And what we find is that where there is seagrass present, there is less enterococci. And so these seagrasses are acting as purifiers and filterers. But the problem is that they're disappearing at alarming rates. So worldwide, we roughly lose about 1 hectare a day. And here I'm showing you the net change in seagrass area across decades, with the most dramatic decrease in the 1980s. And we lose about like I mentioned, we lose about a whole football field a day. So we're not just losing like plants and underwater meadows. I mean, they're not algae, they're actually more related to plants, lilies. But we're losing entire ecosystems. And so some of these, despite all these services that seagrasses have, we are losing them to multiple interacting pressures like bad boating practices, which scar the beds of seagrasses, like sea temperature, increased sea temperature, agricultural runoff, so those excess nutrients, sedimentation, urbanization from reduced light. And another one is invasive species, which we're going to talk about. And so, yes, meet Halophila stipulacea. Yes, it's a seagrass. It does sequester carbon like the other seagrasses, but it has a different ecological footprint. So it's about 3 centimeters, maybe 5 centimeters tall in total in blade length. So it's not like Thalassia that's like 20 centimeters in blade length. And the plant itself has it has a different nutritional profile. It has lower macronutrients compared to the Thalassia, which is highlighted in, circled in red, where you have, there's less percent carbon, less protein, and there is even a higher carbon to nitrogen ratio. So a high carbon to nitrogen ratio is like a woody structure. So trees have a high carbon to nitrogen ratio. And so local herbivores like turtles, they don't prefer it. People have seen juveniles do tend to eat it. So there are adults that it's not in their diet. And Willett in Ambrose 2012 also found differences in these patches with native and invasive seagrasses. So in the invasive seagrass beds, they found a significantly less proportion of juvenile stage fishes. So seagrasses are nursery grounds. Those Thalassia beds that are 20 centimeters tall protect those little juvenile reef fishes. And they also found significantly more crustaceans or amphipods that inhabited the seagrass beds, which makes sense because it's a principal food for these juvenile fishes. So this is what a Halophila dominated bed looks like. So instead of seeing, these waves of of blades, you're seeing changes in waves of light. So it almost looks like a big old carpet has completely sort of taken over in these areas. And so we're seeing this shift, and we're seeing this shift throughout the Caribbean. It doesn't have legs, but it does attach and it creates these tumbleweeds. And as it moves through, so hurricanes really push it through, it attaches to patches of sargassum. So it's really flexible, it can clump together, it can detach, it can float up to 10 days in the water column and then resuspend. And so one of the questions that I have is, where did it come from? This is important for management to figure out, how did it get here? How does it continue to move? And so we know that it's native to the Red Sea and the Indian Ocean. And in 1869, during the opening of the Suez Canal, is when a lot of boat traffics went through. And so you have that Lesbian migration. It's always a hard one. And so Halophila was first documented in Cyprus and Rhodes in 1894. And then in 2002, it showed up in Grenada. And it's moved since then, and I'll talk about it a little bit later. And so a leading hypothesis is that it arrived through boats, transported by yachts. But we're going to use, so one of the hypotheses is that maybe it moved as a single introduction from its native range. So maybe one population went from the Mediterranean to, from the native range to the Mediterranean, and another population went from the native range to the Caribbean. And there might be that, I've seen Halophila attach into these sargassum patches. So maybe Halophila is being swept into sargassum and it's getting pushed in some different direction from the Indian Ocean in some way. Not sure. So what that would look like is that these native populations would be genetically distinct from each other. And so here I'm showing you a hypothetical principal components analysis, which is a statistical method that takes a complex data set with many variables and it distills it down. And so it takes genetic data across thousands of loci and it reduces it into a few variables. And so what we're going to see is that there's a different native that's more related to the Caribbean and a different native that's more related to the Mediterranean. Another hypothesis is that it's an admixture introduction hypothesis, where you get multiple sources leading to the Caribbean population. And so what that would look like is it would be difficult to tease apart who's who, because it would look like one panmictic population. And then the third hypothesis is that there's a serial introduction hypothesis, where there's a stepping stone colonization event occurred and there's a widespread secondary invasion that stemmed particularly successful from a primary invasion. So it's a stepping stone. So the secondary invasion is particularly much more successful. And so this would be where the Caribbean is more related to a population where it historically invaded, and that it would be more closely related to the native population, so somewhere in the Red Sea or Indian Ocean. And if we found this would confirm the leading hypothesis that Ruiz and Valentin in 2004 proposed, that it arrived to the Caribbean from the Mediterranean through boats or yachts, or potentially attached to anchors, which I've seen, or maybe ballast water. And so we figured this out. We can use genealogical relationships because they have DNA. So much like 23andMe, we can use population genetics to trace the ancestry of these invasive species by comparing the genetic relationships. And we can use this by using single nucleotide polymorphisms, which is a change in position that differ across members of the same species. So the beauty of SNPs is that it can be observed variation between populations. So for example, there's a purple and a blue and orange population, and at a particular position, there's a change in the base pair. And these individuals are more likely to share that base pair change with individuals that they're more closely related to. And so you can track these polymorphisms to discern small differences between populations, because they have to be of the same species. And so you can measure gene flow through through FST, which is the fixation index, which describes the degree of population between the populations, so within population and between populations. And so here you can see an individual that carries an a little a allele. So you get one allele from your mom, you get one allele from your dad. And so you can track these changes as you sample the population. And so, so by looking at these genetic relationships, we can try to reconstruct the invasion history. And so I went out and we sampled. Well, I didn't go out to all these places. I had a lot of people who sampled for me. And so we got, we tried to collect Halophila from around the world, specifically across 4 different basins. We have 3 populations in the Indian Ocean, 1 population in the Red Sea, 4 populations in the Mediterranean where it historically invaded, and then 4 populations in the Caribbean. We used a type 2 restriction associated enzyme to sequence 1/16 of the genome. And we ended up, after excluding low coverage samples and low coverage loci, we kept about 94 samples with 524 SNPs at 10x coverage. And so here you see a discriminant component analysis summarizing the genetic structure of Halophila stipulaceae. And each cluster represents a genetically distinct population. And you can see that we have 5 discriminant population clusters. So you have cluster 1, which is the Red Sea population. Cluster 2 includes the Caribbean and the Mediterranean populations. And cluster 3 is the Indian Ocean population, and clusters 4 and 5 is the Mayotte and Mozambique. And so what I find interesting is that when we look at the neighbor joining tree, the clustering is between the newly invaded and the historically invaded populations, where you have as an outgroup, so panel A is individuals from the specific populations, and panel B is from the particular basins. And on the vertical axis represents closeness of the populations, and the numbers on each branch represents the probability of encountering that particular genotype. And so we find that the newly invaded range clusters with the historically invaded range, which is then genetically closer to the Red Sea population. And when we look at FST values, so FST was that fixation index. So fixation index of 0 means that you're identical. And a fixation index of 1 means that you're a completely different species. And so we see that there's a lot of variation between our populations, but where it's closer and lower in value, like we see between Cyprus and the grenadines, we see that it's a lower FST value compared to the grenadines and populations in Timilnadu. So coming back to our 3 hypotheses, our data supports the serial introduction hypothesis, where there's a stepping stone colonization, where there's a widespread secondary invasion that's stemmed particularly successful from a primary invasion. And so we believe that it dispersed, and we confirm, well, the data supports the hypothesis of Ruiz and Ballantine from 2004, which is, I find, pretty interesting. And so understanding where invasive species come from is important, especially in predicting where it might go. We did find that there was low genetic diversity within the Caribbean, much lower than the Mediterranean and the Indian Ocean. And I'll talk a little bit about its reproduction in a minute. But oftentimes, you know, when species get introduced into new areas, they fail. They fail to become invasive. So there are different scenarios of introduced species. So you can have the example of the dune population where a species just doesn't make it. Another scenario could be that there's a non-invasive population. So this population gets introduced, but it remains innocuous or restricted in their non-native range for decades or longer. It could also be that introduced species may establish as invasive populations, where suddenly they expand and they become serious pests, harming economic and ecological biodiversity. And so some of the key questions is, why do some species become invasive while others do not? It could be competitive interactions. So species interactions may promote invasion by having a negative effect on species, like these two ants, where the invasive ant can the red one can outcompete the local native ant population. Or it could be a facilitative interaction, whether mutual or commensal, can also accelerate the invasion of natural communities. For example, you could have a shared pollinator among plants that further propel the invasive species. Or it could be natural or anthropogenic disturbances, so like hurricanes or fires that can promote invasion success, and species interactions may play a minor role in invasion. So one conceptual model that predicts that invasive species are drivers of their own success is the driver hypotheses. So the invasive species can readily displace native species through direct interactions, such as competition or allelopathy, or by enacting in changes in its surrounding environment. And so drivers may outcompete native plants by reducing the nutrient availability. For example, nutrient acquisition in plants can sometimes depend on other microbiota. They may secrete a phytotoxin, or we know that ice plants in California, they secrete salt around their surrounding environment, making it uninhabitable for other native species. So a hypothetical way that we would see that Halophila stipulacea is a competitive driver is that when it grows alone, it's going to grow the same as when it grows with the native species. So this would indicate that no matter whether you're together or alone, you're outcompeting. And for the native species, it would be the opposite, where you would be affected, and when the native species grows mixed with the invasive, it decreases in growth, whereas as opposed to when it grows alone, it has a positive growth. Or if it was some type of facilitative strategy, in the invasive, when it grows with the native seagrass, would have a higher growth rate than when it's grows by itself. So the native species may enhance the success of the invasive species. And same results for the native seagrass, where there would be a decline in the native species when it grew with the invasive, and it would have a positive growth when it grew alone. And there are studies that show that that native species can fix nitrogen and facilitate the available nutrients in the soil, which can be a limiting resource. The third hypothesis, that could be a passenger. We talked about disturbance. Invasive species can succeed because it requires some type of disturbance that removes the native species, especially if you're thinking about a long living species that prevents its recovery. In 2006, Brundo et al. Investigated how disturbance impacted the dynamics of native seagrass Zostera marina and the invasive Zostera japonica. And they found that after a disturbance, Zostera marina decreased in a 14-fold in biomass, while Zostera japonica increased in 11-fold in biomass. So just by creating space allowed for that invasive species to move in. And so there are evidences to suggest that invasive seagrasses may need a disturbance. So maybe the native predators like turtles are eating the native species, which is leaving room for that invasive species to come in. So it's a passenger. And so it's hypothesized that it is a passenger and that it does need a disturbance to invade, because it's only 3 centimeters tall. It's not very big compared to Thalassia, which is a long-living species. Think of like a red forest. So it can proliferate. After a small-scale disturbance, it can quickly colonize an open area. So we did a study in St. John, and it took one dive to remove 1 hectare by 1 hectare meter plot, and by 14 weeks, the entire plot was covered again with Halophila. So it's really, if we're thinking about like a management scale, it's pretty costly. Because it's fast growing. It grows, I'll show you in a second, it grows about 1 centimeter a day in the Caribbean. It has a wide tolerance to light, so it can grow anywhere between 20 meters to 1 meter. And it has a rapid rhizome elongation, so it grows through elongation instead of so much vertical. And so in the Red Sea, it grows about 0.2 centimeters a day, whereas in the Mediterranean, it grows a little faster, like 0.3, 0.5. It really is temperature dependent. They really thrive in hot temperatures. And in the Caribbean, it's around 1 centimeter. And so maybe there's an intrinsic property that's allowing it for it to be so invasive. It also, in addition to a faster growth rate, there's flexibility in reproductive modes. So as you saw earlier in that video, it grows, you saw that tumbleweed, it grows through clonal fragmentation. We still haven't seen the female flower or the seeds of Halophila in the Caribbean, but we do know that there are the female flower for sure, and the seeds inside the fruit are in the Mediterranean and in its native range in the Red Sea and the Indian Ocean. So it's unclear whether it acquired its sexual reproduction in the Caribbean. But since its discovery in Cyprus and Rhodes, so over here is the Red Sea at the very bottom right, and then in the middle is the Mediterranean Sea, and so Cyprus and Rhodes are highlighted in blue. It has moved over 2,000 kilometers, and actually it's now in France. It's now in Monaco. So I need to update this slide, but there was a study done in Tunisia that found a dominant shift towards a Halophila coverage. So in the top left panel, you have it's 2011, and in green, dark green, is Simulosia nodosa, which is the native seagrass. in the Mediterranean, and you have Halophila stipulaceae in light green. And by 2014, it's almost a complete colonization of Halophila stipulaceae. But there's always a small patch of the native seagrass, because we'll find that the same thing happens in the Caribbean. So since its discovery in Grenada, it has moved all the way up to Miami. It's made its way to Biscayne Bay. And it's down in Aruba and Venezuela. And we thought it could only come to Puerto Rico, and it's definitely already traveled. And so there was a study done in Dominica about a long time ago, actually, where in 2008, the native seagrass, Syringodium filiforme, occupied 85% of the area, and the light green, which you can't even really see in the left panel, is about 6%. And then by 2013, the native only occupied 13% in that small patch in the middle, and the invasive occupied about 86%. So even colonizing areas that were previously sand or devoid of seagrasses. And so we decided to try to mimic natural conditions and set up an experimental quest to understand, is it a competitive, is it a superior, is it a driver, or is it a facilitator, or is it a passenger? And so we went to the Caribbean, to St. Thomas, and we chose Syringodium filiforme as its native and prevalent in the Caribbean. And it was definitely prevalent in St. Thomas, where I conducted the study. And we placed 2 standardized cores in each mesocosm. So that scuba diving video where I showed you where I was coring, we grabbed 2 of those and we put them into the middle, and then we filled it up with sand. And then we set up our treatments. So we had 3 treatments. And so on the left is mixed, so how it came, ambient, natural mixed densities. And then treatment 2, which is just Halophila. So we removed all of the Syringodium filiforme, and we left only Halophila stipulaceae. And then treatment 3, we removed all of the Halophila stipulaceae, and we kept only the native seagrass, which is Syringodium filiforme. So we ran this experiment for about 6 weeks. We looked at the difference in shoots, final above ground biomass, below ground biomass, and we ran a 2-way ANOVA. I'm only going to show you the change in shoots. And so what we find is that in the Caribbean is that on the x-axis, you have mixed of the invasive, alone of the invasive, and mixed of the native and alone of the native. And on the y, you have the change in number of shoots. And so we see that Halophila is significantly different than when it grows alone. And supporting the facilitation hypothesis, where the native seagrass doesn't do well when it's mixed with Halophila, but it grows better when it's alone, even though this is not significantly different from each other, because they share the same letter. But we wanted to go and test this out in the Mediterranean. So we got a little bit smarter. We used lift bags because they're actually really heavy. And we included one more treatment, which I won't, which is the reduced density treatment, where we reduced half of the number of shoots for each species. But I'm only going to show you to make a direct comparison, the first 3 treatments. So same thing. So we have mixed ambient. So now we have a new species. We have Seagrass nodosa, which was the same one that we saw earlier in Tunisia. And we have Halophila stipulaceae in mixed. And then we have Halophila stipulaceae alone. And then we have Seagrass alone. So the native alone. And we find similar things where there is a trend for the invasive when it grows mixed to have a larger growth, so change in shoots, as opposed to when it grows alone. But it definitely outcompetes the native seagrass, as the native seagrass does not grow well when it's with the invasive, and it grows better when it's alone. And so we see the same pattern in the Caribbean and the Mediterranean, where it just does better with its friends. And so we wanted to ask the question, we know we have an idea of where it comes from. We have an idea of how it behaves with other native seagrasses in terms of its performance, but could the microbiome be part of what makes Halophila stipulaceae such a successful invader? And so we suspect that microbes may be influential players in Halophila stipulaceae growth and in its invasion. And we think that because we know from studies that microbes help plants acquire nutrients. They help them handle stress and enhance other processes like photosynthesis. So we know that maybe we know that we ourselves are more microbes than actual DNA human, and so maybe there's a correlation. And so we leveraged the study that was done in the Mediterranean to investigate what the phylosphere of both Seagrass nodosa and Halophila stipulaceae look like. And so we initially we started with 40 samples from samples that we collected initially, and we collected at the final time point. We extracted the DNA. We ran it through 16S amplicon sequencing, targeting the V3, V4 region. We then used CHIME to data 2 to assign ASVs. We used the SCUBA database to match, to call the ASVs. But what we ended up retaining was about 18 samples. So that's what I'm going to be presenting today are these 18 samples. So I forgot to mention, because there was 10 replicates, I ended up making about 4 composite samples of all of the 10 replicates because I didn't have enough money to sequence every single tank. So I ended up having to make composite samples. But then once we did the DNA extractions of those composite samples, that's what we used as a way to have a complete data set where we have the microbes, the morphometrics, and the biochemical analyses, because we also took chlorophyll A, chlorophyll B, total chlorophyll, MDA, and hydrogen peroxide, which are indicators of stress for the plant. And then we have all of the morphological features that we, covariates that we have. So the number of shoots, the above ground biomass, internode length, below ground biomass, and then what we had in the soil. And so alpha diversity between the native and the invasive seagrass were similar, but community composition does differ. So there was no significant difference between the alpha diversity. However, in beta diversity, where you see at the bottom, you have the circles are the native seagrass, the triangles are the invasive seagrass. There is a significant difference in the community in terms of species. And next, we wanted to look beyond diversity metrics and ask, okay, who's actually making up these communities? And so across the samples, we have proteobacteria, which is dominated the phylosphere in accounting for about 72% of the sequence reads. But when we look more closely at the relative abundance of each individual taxa, we see stronger differences between the native and the invasive seagrass. So for example, Cimodosia nodosa had a substantially higher relative abundance of bacteriota, and several bacteria taxa that were absent were much less abundant in Halophila stipulaceae. So this tells us that there's two host differs in particular bacterial taxa that dominate on their phylosphere, because I only took the microbes of the blades, not the soil, not the roots. And so when we ran Maslin 2 to identify which bacterial taxa were different from each other, we used the reference condition as the control for Maslin. So that's how it grew in natural mixed treatment. So for Cimodosia nodosa, we're asking what changes are there in the bacteria when the native grows alone or when it grows in reduced mixed density compared to when it grows fully mixed with the invasive. And we found that Disopholbus, Bermanella, and PS1 clade were significantly positively associated with Cimodosia nodosa when it grew alone compared to when it grew in mixed conditions. And then for the invasive seagrass, Halophila stipulaceae, we're making the same comparison, but using the invasive in the natural community as a reference. And we found that bacteria like Photobacterium were significantly negatively associated with the invasive when it grew alone than in mixed conditions. And that there was a significantly positively association with Rhodobacteriaceae when it grew alone compared to these mixed conditions. So the key point from this is that there are bacteria that are different when we grow together and alone. I will say we cannot assign specific functions, so I put these relative functions here, but you have to remember that it's only 16S data. So it's not going to tell you much about function, but more about abundance and how many is there. And so now we're looking at the microbial community composition of the Bray-Curtis beta distances with the biochemical and morphological covariates using the Bray-Curtis distance. And so we see that the triangles are still the invasive and the circles are the native. We still see a clear separation between the two species, although there's more variation, or there's less variation in the native species. And for hydrogen peroxide, which is a proxy for cellular stress, it was significantly associated with the native species, explaining about 61% of the variation. And the internode length was also significantly associated with the community composition of the native seagrass, explaining about 49%. Whereas in contrast, the number of shoots for Halophila stipulaceae, even though it was not significant, there was a weaker association, but there is a non-significant trend towards the invasive seagrass. And so we wanted to look a little bit further to answer the question about the core microbiome, and we looked at least 80% of the samples across both species. And we identified 53 shared core taxa between both native and invasive across all treatments. And we also found that Cimodosia nodosa had a higher core microbiome than the invasive, with only 24 genera. And so looking at still taking a look at the core microbiome, we took it a step further to look at the relative abundance of those core genera across treatments. So on the top, you have native mixed, you have native reduced mixed, and you have native alone. And then on the x-axis, you have all of the taxa. And so for native Simulacea and Alosa, the microbiome in the reduced look very similar to when it's native alone, and native mix looks completely different to when the native is alone. So it's possible that the presence of Halophila stipulaceae is associated with the stronger shift in the native's core. It's unclear because I only took the phylosphere. I didn't take the roots, I didn't take the soil. I just sort of scratched the surface and asked the question, and we do see. So it's not, I can't say this is fact. I can only say that there's a trend. We can see maybe there is a relationship. And so the inverse happens with the invasive. So the invasive showed almost the opposite pattern, where its bacterial communities were more similar between natural and reduced treatments, while when it grew alone, it's very distinct from when it grew mixed. So the conclusions that we can make from this microbial diversity in this competition experiment is that even though alpha diversity did not differ among treatments, but they differed across taxa, and that the core microbiome patterns do shift when both the native and the invasive seagrasses grow alone and or together. So we do see differences in relative abundance for those treatments. So what makes Halophila stipulaceae an invader, an amazing invader? Well, so putting all the pieces together, we think that Halophila stipulaceae is a stepping stone colonizer. We think that it was from the Mediterranean, and it's really established and expanded throughout the Caribbean. And what's particularly interesting is that it isn't necessarily replacing the native seagrass outright. It's growing alongside of it. It's growing in areas that it once were devoid of seagrasses. So, and we see that in both the Mediterranean and the Caribbean. But there is no doubt a change in the ecological services that it has. And so one possibility is that maybe the plant's associated with microbial community involved. Maybe it's other intrinsic properties. Its genome is huge. I mean, I just spent money on sequencing the genome because we had to sequence it again because it was, oh my gosh, I should have checked the size. But it was 15x more than what we expected. I mean, I wasn't expecting that large of a genome. So there could be other reasons. There could be epigenetics. It does grow faster in the Caribbean. It's bigger in the Caribbean. Its phenotypes are different. Whereas in the Mediterranean, the patches are very ephemeral. So they grow when the temperature is nice, and then when it gets cold in the Mediterranean, because it does get cold, those patches tend to stay put or even decrease in size. And so I have I have students working right now on do manatees eat it? We think that they don't. Just like the turtles, the adults don't eat it. The juveniles do eat it, and there have been necropsies on manatees in the south of Puerto Rico that have showed Halophila stipulaceae in the gut. So the lack of predators, the huge genome, the ability to grow fast in temperatures, it's and not necessarily kill the whole native seagrass when it's colonizing a place. So, yeah, so it's a really cool species. I think there's still a lot to uncover. We're still trying to find the flower, the female flower. You know, how can it be that it's just so, how can vegetative fragmentation be the only way that it really grows? So I find this species really awesome, and really we have to embrace it. I do know that it is affected by light, right? Because it's a plant. And so when you get these huge sargassum inundation events that we've seen recently, it does wipe it out. There is like no Halophila at the bottom. I mean, there's nothing. I mean, there's like, it looks like a graveyard because there's like dead octopus and a bunch of like urchins. Well, I guess tests from urchins, but that's all that I have. Thank you so much for listening. There's 10 minutes for questions, so I'm happy to answer any questions. Please follow us on Instagram if you want to know more about my students and what they're doing, because we're doing a whole bunch of other stuff. So yeah, one person's looking at manatees, but another person's looking at, can we take the sargassum, use it as biochar and put it back into seagrass beds? I have another student looking at how urbanization from shading affects the gene expression of Thalacea. So I'm not necessarily working with Halophila, but the native species and other native seagrasses, and resilience and adaptation is something that I'm also really interested in. So yeah, thank you for listening.